Yes, thank you for introduction and for your invitation to this wonderful online meeting. So um, my name is Zhang, the currently work at the University of Queensland. So today I'm pleased to share the research topic of my team in the University of Queensland, work on the background-free luminescence biology and imaging using the non-lifetime responsive probes. So my talk today will include these 4 parts. First one, I will introduce some background information, including the background information of the response probes and also the active biomolecules, and how can we design the response probes for the biomolecular sensing and imaging. And in the second part, I will briefly introduce some of the examples of our research on design of the nicely chelates and the lutetium dyes, iridium dyes, and also the unconventional probes for the background-free luminescence detection and imaging. And then third part, I will show you some of our efforts to translate our research outcomes to the clinical, preclinical, or commercial products, and then we'll make conclusion for the for the talk. So as we know, our health, the human health, is heavily affected by the surrounding environment. For example, under the stress, the growth of the normal cell will be affected, and the damaged cell will be produced. So the damaged cell can either be cleared from our body, so the immune response, and or they will undergo very fast proliferation to be the damaged tissue. So if we can track the fate of this damaged cell, so we can get to know the information of the damaged disease, the progression of the disease, and how can we track the fate of the damaged cell? Normally, what we can do is detect or monitor the biomarkers, so the biomolecules inside the for example, the biological fluids like the blood, like the urine, or do a cell test, or the in vivo test, and of different target analytes of interest. We are particularly interested in this kind of the biomolecules like those IS, RCS, and the RCS, and the antioxidant, the amino acid, and some such as so the protein DNA, like the macromolecules. So here this table shows us some examples of the active biomolecules. For example, those normally include the hydrogen peroxide, the single oxygen, the hypochlorous acid, the hydrochloric peroxide. And we know this superoxide here is the first those to be produced in the body, and the superoxide can be converted to the hydrogen peroxide, and the hydrogen peroxide is much stable, can react with the metals like the ions to produce the hydroxyridical, and also with the chloride to form the hypochlorous acid. So why we are interested in this kind of the biomolecules, just because they are it's very hard for this kind of biomarker detection. It's very small sizes and small nanometers, and also it's invisible hide within normal tissues. And most importantly, these are highly reactive. That means once production, once activation of this kind of biomolecules, they can react with other biomolecules nearby and cause damage to other tissues. For example, the hydrogen peroxide enzyme introduced is the most stable one, but the diffusion distance is just around 1.5 millimeters. And for some others like the hypochlorous acid, like the hypochlorous acid, the hydroxyridical, the diffusion distance is even shorter. It's just around 20 micro, so just around size of the size of one cell. So and actually this kind of biomarkers are very important for us. For example, in the oocyte, the macrophage will express high levels of the and those to kill the bacteria. But if the all bacteria has been cleared from the body and the high levels of those kind of frequencies will be this kind of the reactive biomarkers can react with other biomarkers in the, for example, the membrane, cell membrane, the protein, the DNA, to cause the damage of that kind of the damage of the structural damage of that kind of molecules, and cause the disease, like the most commonly the inflammation. So and for some other diseases like the cancer, if we quickly increase the concentration, the levels of this kind of biomarkers, the cancer cells will be killed. That's actually that's the principle for chemistry, for example, for most of the cancer therapy, like the photodynamic therapy, radiotherapy, and also some of the chemotherapy. So research objectives of my team is to find out and quantify these biomarkers, so the biosensing, and we also use the biosensors to monitor the invisible biomarkers by our eyes. So the imaging, and we also put some biomaterials to regulate the levels of these biomarkers inside of the body for treatment. So here shows the research topic of my team here in UQ. So we are working on create some optical biomaterials, including the phosphorescent metal properties, the transition metal and organic dyes, the nanocrystals, like the upcoming nanocrystals, the quantum dots, silicon, the neutron hydroxide, and polymers. So, and then we are using this kind of the materials to create some, the biosensors, for example, the probes for the detection of the particle analytes in in cells, in tissues, and in vivo. And we also create some materials to regulate the concentration, the levels of the biomarkers inside the body for the treatment, like the kind of treatment, inflammation treatment. And recently, we are also interested in using this kind of the sensors for some other applications, like the detection the indicator in the faults for the fault safety and quality detections. So what kind of the biosensors or information used in our research? We can see here. Here shows the principles of our responsible probes. So, for example, if we want to know the information or levels of this type of analytes in this biological samples, what we can do here is we have these probes to be designed, and normally the probes has 3 parts. We can see here the single units, the positive, and also the coupling units, and also the spacer here. And after binding or reaction of this target analyte with this probe, we can see the signal, the fluorescence or emission signal will be changed, and we record this kind of changes to quantify or to get the information of this analytes in the sample. And we're using this kind of signal change for the cell study, for the tissue study, and also for in vivo studies. And I introduced the signaling units is the most important part of the responsive probes. So normally, the probes are designed based on the the fluorescence dyes and organic dyes. Like this table here shows some of the dyes, like the formalin, the FITC, the bodipine, the glutamine, 7 dyes, and also some neon-infrared 2 dyes. And previously, we also have some research work on the organic dyes. And here just to show one example, we have this probe for the nitric oxide detection. You can see here after reacting with nitric oxide, you can see the rotation of this bond will be blocked, inhibited. So that means the quenching fluorescence of this probe will be switched on, and we use this probe for the nitric oxide detection in the inflamed cells. We can see here we get the LPS stimulated the inflamed cells, so we can see the nitric oxide levels is increased in the inflamed cells, and we confirm the probes can be activity in the cells, in endosome, in mitochondria or other cell organs. And when we are doing the field research, we find a problem that is the the autofluorescence, the background signal that is, especially when we are doing the when we are doing the more complicated biological samples, we can see the high signal from the autofluorescence, and also that scattering. So normally, what we can do currently is using the filter that's both the microscopy or spectrometer when using. So we, for example, if we are using this filter, we can see only the emission from this range from 350 to 400 can be transparent, but other signals will be blocked. So that means the signal we are recorded just from very short range of the emission wavelength. But the problem is where the autofluorescence background signal can be from the UV to D range. That means the signal we recorded, even though we are using the filter, always mixed. The signal always mixed from the our probe responding to the target analytes, and also the autofluorescence and scattering. So here shows to a stage where we used to remove the background signal for the background free luminescence detection. Imagine here, the first one is the time-gated luminescence detection. Here we have here we can see here we use the lifetime because the lifetime of our probes are very long, but for the lifetime of the background signal or the autofluorescence dyes, the lifetime is very short. So here, once the post excitation, we can see here all the molecules, all the dyes, all the fluorescence dyes will be excited, including the autofluorescence dyes, and also our probe will be activated to the excited state. And for the autofluorescence dyes will undergo very quick, very fast decay to the ground state. And so after this time, we start to record the signal, and this signal, in this range, separated from the our probes responding to the target analytes. So here is just choose some example here, so this is an image from the optic filter. We can still see very strong background signal, and for the time-gated one, we get the image of the target state. We get very high signal to noise ratio. Second one is the up-conversion luminescence detection. We can see here, different from the the traditional conventional fluorescence process. It's from high range to low range emission process. Different from this, the up-conversion one can be excited by the lower energy excitation and get the higher range the emission. So with this one, so almost all of the autofluorescence dyes will not be excited. So so for the first stage, we're using the time-gated one. We can see here we did the long-lifetime dyes. Here we can use the transition metal complexes and also nitrogen chelates. So for transition metal complexes, we normally use the same like dyes. We can see here we just need to modify the structure of the pyridine ligands and get the probe for the nitrogen chelates. We can see it's a little bit different just because the emission of the nitrogen chelates always from the metals, and the absorption is from the the antenna from the ligands. So we just need to modify the structure of the antenna to get the probe, get the energy transfer from the antenna to the metals activity or inhibited. So that's the principle for the design of the probes. And here I have a table to show you the difference between each of the fluorescence first. We can see here for the transition metal and nitrogen chelates, we can see very large Stokes shifts, and also the very good for stability, and most important, they are long lifetime for the time gated luminescence for remove the biomarker signal. So previously, or early stage, we designed some of them. We have some of the nested kits and the probe. For example, this one was designed for the hyperprocess detection here. This one is a pincher after cleavage of this bond. Of this bond, we can see the emission will be switched on, and very long lifetime we see here more than 1 millisecond. Because then we get the time gated luminescence for the hard-core detection quantification, and we also the imaging will purify the signal from the probe responding to the hard-core. But we also get the problem that is, even though we get the long lifetime microseconds to milliseconds, we can only the accuracy can be affected by the UV lights. The UV lights has the short shadow penetration depths and also the higher toxicity. So we cannot be used for the flow cytometry detection for the large scale cells detection, and for the others like the stream iridium complexes, we can see the lifetime is more than more than 100 seconds, and it can be excited by the visible or near infrared light, and the site shows high penetration depths and also lower toxicity, and then they can be used for large scale cells detection. So we then focus on using the exoskeleton and iridium dyes. I think I will show you some examples of the exoskeleton and iridium dyes for the biomarkers detection. I think the first the active biomarkers detection biomarkers responsive probes based on the exoskeleton complex was designed by me in 2010. I think 12 years ago, and this probes you can see here can detect the nitric oxide after the action with the diamine group with the nitric oxide to form this emissive, the products. We can say the phosphorescence emission, will be switched on, and also the electron chemical will be switched on, and we get double channels for the nitric oxide detection. And later on, when we compared this probe for the nitric oxide detection in the biological samples in comparison with the commercial available product, the probes, the DAF FM, we can see here our probes has comparable or even better performance, in the detection of the nitric oxide in biological samples. And currently we work with the medical institute for translation this research to the clinical tools for the detection of nitric oxide in the cardiovascular disease patient samples. And the other example I want to share here is the probes for the formaldehyde detection in here. We call this one the dual K and local probe just because the probe the emission of the probe can be switched on after reaction with the formaldehyde in the acidic condition. As we know, in fields only the lysozyme shows the acidic condition, so we can use this probe for the lysozyme formaldehyde detection. Here shows the structure, chemical structure, and the sensitive reaction of the probe with the formaldehyde. And we can see here the probes, the quench of the fluorescent luminescence is because the photo-induced electron transfer from metal to the quencher here, DMT here. So after cleavage of this part, we can see where typical metal to to ligand transfer process of the neutron complex. And when we are using this probe for the biological studies, we can see here even the way for the blood for the formaldehyde detection in the blood, we can say even though we had 10 times dilution of the sample, we can still get a very strong background signal from the serum samples, and we cannot get any information from our probes responding to the to the formaldehyde. But if we introduce a time delay, we can see the background signal was completely removed, and we get the signal of our probes responding to the formaldehyde in the serum samples, and we get the standard curve for the quantification, and get the concentration of the formaldehyde in the serum is 100 micromoles, and we also get the formaldehyde in the mouse organs are less than 0.5 millimole per gram, and we also tested we also confirmed the probes can be used for the formaldehyde detection in the cytosomes. So here, as we know, the tumor has higher activity and also lower pH, so the probes can be used for the formaldehyde detection in the tumor tissues. You can see here we inject the probe to the tumor, we get the very strong signal from the tumor, and we also get the X vivo imaging. We get strong signal from the tumor tissues, and that's the story of the reduced dyes. And then we also had some steps for the for the hypochlorous detection using same principle. We designed this probe for the hypochlorous acid detection in using the iridium dyes. Because here with very strong signal after reaction with the hypochlorous acid and very specific reaction. And when we are using this probe for the for the hypochlorous acid detection in the liver injury, we had the ischemia, the perfusion treatment of the liver, and we get the signal here is much stronger than the control group. So and we also find the difference that we are using a 2-photon excitation, 2-photon microscopy, we still get very strong background signal. So we moved to the background phase imaging using time gated one. We designed this probe for the safety detection. So we can see here after reaction with natural group with the detection to form this emissive complexes, we get the specific reaction, and we get the the time gated luminescence spectrum, and the standard curve for the safety detection in the in the serum is around 250 micromolar. And we also demonstrated that our probes can be used for the for the safety detection in inflamed cells. We can see here for the inflamed cells, the signal in intracellular signal is much weaker than the control group, and that's because the cysteine has been consumed for quenching those inflamed cells, and also to production of the hydrogen sulfide in the inflamed cells, as has been confirmed in our another paper published in 2018. So in the time-gated luminescence detection, we confirmed that after 6 or 8 nanoseconds delay, we can see almost all the background signal has been removed, but for our probes, for more than 40% of the signal will be retained. So we get a very very high signal to noise ratio with the time-gated luminescence detection. So we have also designed some probes for the time-gated luminescence detection. For example, this one was designed for the biocells detection, as well as biocells normally contains cysteine, homocysteine, and GSH. And if we want to test the total amount, and also or if we want to test, if we want to get the concentration level of each of them, we need one probe for one purpose. But for this probe, we designed this probe for for multiple purpose. We can use this probe for the total amount of the biotech detection with the time-gated luminescence detection, and we can also discriminate the cysteine, homocysteine, and GSH. So so recording the color change, the fluorescence, the emission color changes of the sample. So here, that's the table show us currently the probes are available for the biomarkers detection. The available probes are showing here the red color, and the black color one shows here the probes are developing. And we also summarized our time-gated luminescence detection imaging sensor in this account. So if you are interested, you can click this account. And we also designed some, as I also introduced the another study, the upconversion luminescence. We designed some of the the probes for this responsible probes for the biomolecular detection using the upconversion nanoparticle. So we here we use this to them as the acceptor, and the upconversion nanoparticle as the donor. We build this ion transfer system just because the the absorption of the Jerusalem dyes overlaps with and where with the emission of the upconventional particle. We can see here after the reaction of the nanosensors with the hyperprocessing, we can see the absorption of the Jerusalem dyes will be decreased, and the emission of the upconventional particle increase. So we use this kind of the upconventional luminescence increased for the detection of the hyperprocessing in the cancer cells and also inflammatory cells. We can see here, then for the inflammatory cells, the upconventional signal and also the signal from the Jerusalem channel are much stronger luminescence than the control group. And we also use this probes for the detection of the hyperprocessing production in the inflammatory tissues. So the third part, I will just briefly introduce some of our efforts to to translate the our research outcomes to the preclinical tools or market. We are currently working on using the probes for the determination of the redox biomolecules inside the macrophage, and we want the answer the question is how to detect the highly reactive biomarkers inside the cells, and also how to get the quantitative connection relationship between the redox biomolecules level and the macrophage polarization, and also the inflammation progression. So we want to build that kind of relationship. And also we are also interested in trust our probes to the commercial products, to get to be available for more researchers in worldwide. So I will get a quick conclusion. Actually, we are working on the optical materials for the sensing, imaging, and also treatment. The treatment part I haven't introduced. I didn't introduce in this presentation. We designed and prepared a service of the responsive probes, for the detection of the active biomarkers in vitro and in vivo, and we also had to switch to remove the background signal for the background-free luminescence detection and imaging. For example, in the biological flights, like the human, like the serum samples, and also for the imaging of the biomarkers in vitro and in vivo. Then we also had some materials for designed for regulating the intracellular biomolecular levels for the treatment. That's something about the nanomedicine. And we are currently were interested in translating the research outcomes for the clinical tools and also the commercial products. So so I would like to thank my teams in UQ, some of my students here, and for some my students here, and also my collaborators in US, in Australia, in China, India, and Sweden. And also I would like to thank the funding support from the Australian government, the ARC, the HMRC, the UQ, and the previously from my Corey, and also the SFC, and also thanks the facility, the equipment support from the AFF, and also the CI, the IMB, Coffee, and also the CMM. And that's all my presentation. And thank you for attention. And down here is my my contact information. So if you are interested in my technology, and if you want to do a collaboration, or if you have questions, just please find me. You can find me by email, Twitter, or WeChat here. So that's all my presentation. So and I'm ready for any kind of question.